Good morning, everybody.
Bonjour à tout le monde.
Yeah, welcome to room 4.
Bienvenue dans cette sous-commission 4.
And this is an open room.
C'est une
Topic
commission ouverte pour
with
les sujets.
various issues, whether it's science, how do you funding,
Il y a des sujets qui nous sont donnés pour lesquels nous allons devoir discuter.
How do you get on the expeditions? What does it take to become a marine biologist? How do you go diving? So just
on va discuter sur comment faire pour devenir un microbiologiste pour ceux qui vont, comment faire des plongeons en profondeur, des explorations en profondeur.
So basically, we can start. If anybody's got a burning question,
je pense que nous pouvons commencer. S'il y a des questions,
please feel free just to start the question.
Sentez-vous à l'aise pour
My name
poser des questions. Moi, je m'appelle
Tawfiq Samai.
Je m'appelle
I'm with the Department of Environment, Forest and Fisheries, situated in Cape Town, which is a government department.
Nous sommes basés à Cape Town,
is Roxy,
département du gouvernement.
who's also with me in this chat room as well.
Il y a aussi Roxy qui est dans cette
please feel free
commission. Alors, sentez-vous à l'aise.
Ask your questions.
Et poser des questions si vous en avez.
There must be a burning question. Anything.
Je pense qu'il devrait y avoir des questions, quelque chose.
Hello.
Hello.
Is it? It's Tim Natkemper speaking. I have a little problem because I hear the French translation all the time, who's doing a great job, but I cannot switch it off.
En français qui parle, mais je ne sais
Because
pas
it's a little bit confusing.
Ça me dérange un petit peu.
Thank you, Tim. I'm going to be contacting the tech team to find out how we can sort that out.
Yeah, okay. Thank you.
Okay, thanks. Thank you, Tim.
Yeah. Maybe I can just start with this very general question. I'm a little bit of an outsider here because I'm a computer scientist, and our contribution is this software platform Beagle for manual or semi-automatically annotating marine images and video. Maybe I could just ask, this was we gave a presentation on Wednesday.
Présentation ici.
What is your impression here in this chat room? What kind of role will image or video play in the future? What are the trends you would expect?
Et les outils qui sont utilisés ici.
Yeah,
quelle est la tendance, votre impression?
Thank you, Tom.
Merci,
well, in terms of the imaging software, I don't think that we will be able to effectively.
En termes de l'imagerie, pour tous les autres softwares ici, je ne sais pas si nous serons en mesure également de.
Without the video software. And in future, it will actually play a much more important role as well, especially if it's got an artificial intelligence a part with it as well, where we can actually, teach the software
Bon, je
to
pense
actually
qu'on va
identify
chercher à voir comment
various
faire pour
structured
certains
images
softwares
on the videos or the photos that we've got as well.
pour les vidéos et les photos que nous avons reçues ici.
I mean, we've been using, the old style.
Nous avons utilisé
The VLC and whatever as well.
types de style.
And
On va voir ici, c'est pas tout ce qu'il y a.
and then there was two other softwares.
Il y a aussi deux autres softwares.
The Beagle software, and that is where we actually
Le software Beagle,
the
je
Beagle
pense
team
que
as well.
vous connaissez,
Because I mean,
avec
it's integral
l'équipe de Beagle,
in
avec laquelle
terms
nous
of
travaillons également.
what we do. It actually speeds up the analysis time as well. Because I mean, you can't spend 5 or 10 hours looking at video
Avec
footage
l'analyse de temps
and
ici,
then do the
on
old
peut voir
manual styles where you've
tous
got
les
a
styles que nous
software where you
avons.
can just look at the things. It makes it much more easier.
On regarde
The
l'équipe
interactive tools
avec
are
nous
there,
discutons pour voir
to speed
comment
up the
rendre les
time
choses
from
encore
10 hours to 2 hours, whatever. So and it makes a huge difference to them.
voilà,
Yeah. So I mean, that is my take on it. It's definitely an integral part of future work.
Okay, thank you. I don't know if I understood everything, because it's still this French translator online all the time, and it's hard to unmix your two voices inside my head. But nevertheless, thank you.
Yeah. I mean, Tim, I mean, the one thing is now as well is that, I'm so glad that that one of the talks were on Beagle as well. And it is open source software as well. And I mean, for us, it's a start for, where we can actually start collaborating with you guys as well in terms of, making our work easier. In terms of future endeavors as well. Yeah. Thanks, Tim.
Hi, Tawfiq.
Yeah.
Sorry, it's me. It's Tanya. I just, there was a little bit of confusion with our French interpreters. So they will not be translating everything you say from now. So just, guys, if there is a question in French or if there's something that you didn't understand, then the French interpreter is there to translate for you. Thanks.
Thank you, Tanya. Are there any other questions? No other questions?
Hi there, Tawfiq. I'd just like to mention how the word cloud works. In the chat box, you should see a message saying word cloud with a link. If you follow that link, you should be able to submit a word or a phrase anonymously, and it should appear on the shared screen, and we can discuss that. That's just to all the attendees in the room.
Okay. Thanks, Roxy.
Tawfiq and Roxy, sorry, I'm still here. If anyone is interested, we can play that little video from the Zico Museum. Wayne and Albi's video. And it will just give people a good sort of overview of the importance of museums and reference collections. If there are no more questions, I'm not sure. So I am here, just WhatsApp me if you guys need me to play it.
Thanks, Tanya. I'll let Tawfiq decide.
But otherwise, yeah, like Roxy said, you guys are welcome to move between the other rooms. If you have any burning question, come back to this room, or if you think of something that you haven't been able to communicate with us during the workshops, then also feel free to do that. But Tawfiq and Roxy, I guess this is just a room where everyone's going to come to eventually when their session ends.
Yeah, that's correct, Tanya.
Okay.
And maybe we can just play the videos along.
Okay. This will stop other screen sharing. I can't actually. Tawfiq, you're gonna have to play it from your side.
I have stopped the screen share there, Tanya.
Stop it. Yeah, I don't think I started it. Robin?
I mean, this should have been the more fun chatty room, isn't it?
Yeah.
Yeah, Tanya?
Sorry, I was just trying to share that video from Wayne, but so if I share my screen, it's going to say that what says that everyone else will screen sharing will stop, and I don't want that. I'm not sure what's happening in the other rooms.
Yeah, I've jumped through the other rooms and I see people are, I think Loren's showing his presentation and.
Okay. So I don't want to fiddle on anything. Do you mind just waiting around here in this room?
Yeah, we told you in this room.
Because I'm trying to Tawfiq, I'm not sure how am I going to do that closing or how we're going to do it, because I've got messages and emails and Word docs everywhere, so I'm not sure. And you said that you sent me something too.
No, don't worry. We're just going to keep it simple, eh?
Okay. So I'm just going to
just a blah 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 stuff, and keep it simple. One paragraph. You know, what's the next step? That's
okay.
And so, then you can thank all the other people and
yeah,
you know the companies and the institutions and.
Okay, maybe we must just discuss this first before we, like, maybe during the break or if there's time in this room.
Yeah. You stressing again or what?
A little bit, yes. But let me just sort it out quickly, because I see we still have in this room. We've got Matthew here. Matthew? Hi, Matthew.
Hi, Matthew.
Hello.
How are you?
I'm good. How are you?
Ah, good. You're sitting quietly there in the background, eh?
Yeah, so I signed up for the workshop when I saw it come around, just
cool,
out of interest. And then as we went through the sessions for the week, I realized that these are a lot of things that I actually haven't engaged with myself. I am currently doing my master's at UWC in conservation biology. And I am currently part of a quantitative snake ecology lab. So the marine environment is still very much a new frontier for me.
Well, the one thing is that, you know, you like snakes. Why don't we pick up looking at sea snakes? I mean,
that is exactly what I was thinking.
Around here, whatever as well. It would be nice to actually tap into that domain as well. Ezekiel Museum's got a number of not dead, curated
Mm-hmm.
Specimens.
Mm-hmm.
But I mean, I've never seen a sea snake.
Yeah, so all the
I mean,
records
we've
that
dived
we
all
have
over.
of them
Yeah.
are just, you know, kind of incidental. They were washed up, or maybe they were part of bycatch or something like that. And so I thought, you know, as we were going through the different sessions for the week, how can I kind of integrate what I've learned here with what I already know? And I would in future, if the opportunity presents itself, yeah, I wouldn't mind working on a project using Bravs and other types of video monitoring systems to help, you know, try to untangle some of those questions and mysteries around the ecology of our sea snakes, and you know, how often are they on our coast? How long do they spend in at different depths? You know, do they ever come into the intertidal zone? Do they not? Where do they hunt? All these kinds of things. So
exactly, yeah, they're very, very elusive. I mean, there's probably one or two places where they come to the intertidal, but you know, I don't know who saw it, but somebody saw it. But that's it. Who you working with at UWC?
I'm part of Dr. Brian Maritz's lab.
Oh, it's Brian. Okay, cool.
Yes.
Cool.
Yeah, so yeah, it was interesting. It was interesting, and I learned a lot. Obviously, at some points, I felt like I was a bit out of my depth, and decided to just be a fly on the wall and kind of listen in and take what I can. But yeah, it's been really interesting. I suppose one question I would have is with the video sampling. A lot of those cameras always go down to the bottom, and you know, you keep them at the bottom and then you kind of survey from there. Do you have other setups where the cameras are at various levels in the water column? So, you know, maybe surface level and then 5 meters, 10 meters, 20 meters, or are they always designed to just be, you know, sitting on the bottom and then surveying from there, because now it's baited and the fish are coming through, or any other organisms?
For the work that we do, which is actually looking at the benthos, the bottom zone, and that is why the cameras actually sit at the bottom, so that we can get the interaction between, for example, fish and reef habitat. What type of fish are at different depths or whatever as well. But I mean, that doesn't mean that you can't design cameras, you know, that sit in the water column as well. You know, but you just got to be saying whether it's going to be drifting or whether it's going to be hanging from a rope while you're sitting on a rubber duck or whatever as well, with different cameras at different depth intervals. You will never leave a camera system at the surface because somebody will just snatch it, steal it. So yes, even though it's benthic orientated, you know, there are designs and stuff and whatever as well where you can actually do pelagic or midwater designs as well.
So have you ever, in your experience, have you ever come across any of those sea snakes, you know, just by chance during a sampling trip, or while you're going through some of the data that you've now collected, and you see, oh, you know, one swam into frame? Or
never. You know, it's like you're asking the question whether people see the snow petrel. I mean, you can either you'll see the snow petrel if you go to an altar, but not a lot of people actually go there. So it's the same thing as well, is that these things are there. But I mean, we've dived a lot along the coastline. But I think you probably need to dive 10 times more to be able to really see these particular things as well. They are very, very, very elusive. It's not like Australia, like the Northern Territory in Australia, where they're just all over. Not these ones, they're probably shy as well. So I don't know.
Yeah, sampling for snakes on land is already quite a big problem in terms of just sampling effort, and then the actual number of individuals that you end up finding, it varies so much. So I can imagine with the ocean, and with the ocean being so vast, it's where do you even begin, you know? Where do you start looking for them?
Exactly.
I've just seen Robin's got her hand up there. I'm not sure if she wants to add.
Hi, Robin.
Hi, everybody. It's me. I just wanted to say, I find it very inspiring, Matthew, that you're actually attending this workshop. I think some of the best research actually comes out. You know, we get stuck in, we're working on terrestrial stuff, you know, we work on terrestrial, we work on marine. We don't really branch across the different scientific fields. So some of the best discoveries and things like that actually come from people who look at other fields to actually, you know, innovate or make new designs or things like that. So I think that's really inspiring, and I'm glad that you actually attended this conference. And then just to say as well, like with the baited remote underwater videos, all of that, but they are on the ground, but we also use ROVs, so remotely operated vehicles. So if you're on a small vessel or on a big, you can actually release like a little, it's like almost like a remote car. Like, you know, when you drive a little remote car, you can actually release one of those ROVs from your back of your ship, and you can actually drive it. So you pilot it and you go across the benthos and move across. So that's another thing as well. They're not always stationary. So yeah, that's just another thing I wanted to say.
Sure. So it's like drone technology just for the ocean. Having little marine drones.
Yeah.
That's really cool. Do we have that, or do we have access to that kind of equipment in South Africa? Do any of our institutions or labs make use of those remote controlled ones as opposed to just the browse? Or
we've got a SIAB. It's got a Sea Falcon ROV, and it's actually being used throughout the year on various projects. Yes. So if you do want to use it, you got to book it. So there's a big demand for it as well. The Department of Environmental Affairs is also looking into buying a smaller version. But there's also some talks that if we get the new vessel, there will be a ROV on that particular vessel as well. But those are all talks for now. Yeah.
Okay, so if you thinking of doing research in it from an institution point, you'd have to approach the department and arrange, you know, like hiring one of these vessels or getting onto the vessels and then just going out as a member of the crew and working from there, or do you have to kind of try and get your own at your institution and then?
No, you know, the department's got a number of vessels. I mean, we've got something like 3 big vessels, and then SIAP has got these smaller vessels that can also go down to about 40 nautical miles, I think. And they've got 3 or 4 of those particular vessels in Durban, in East London, and Algoa Bay, whatever. So basically, if you collaborate with some of these institutions or get funding through them as well, you can actually ask the ship down. The same with the department as well, is that we've got a number of cruises going out as well. So you piggyback on those cruises and partner with, not partner, that's the wrong word, collaborate with somebody in the department or whatever as well. So basically the researcher don't pay for the funds using the department vessel, because we are going out there already as well. So, I mean, that has been covered by the department. And then there's also these other cruises as well with the Agalas too, that certain institutions also take out, but that is being paid through, for example, DSI and the Department of Environmental Affairs compensate some of the funding for that as well. So yeah, you don't have to have the 20 million to hire this. It's actually free. Yeah.
Okay. Robin, if I can ask you a question.
Oh, of course.
I know you did a talk, was it yesterday or the day before, where you just spoke about you know, some of the curation and working at the museum and so on. Thank you so much for that.
Oh,
from your point, have you come across any of the sea snake specimens? How many do you guys maybe have? And in terms of like just going out and does the museum organize any kind of exploratory trips just to see, you know, like on a regular, like annual basis, just like a survey, general survey to see, you know, what are the status of some of the different species around the coast, and are there perhaps any new species, or do we see new migratory species coming in and leaving, or anything like that?
Yeah, so to answer your question, I haven't been based at the museum for very long. So I haven't had, and we in the process of moving all the collections. So recently the museum was renovated, or especially the marine biology department. And so right now we're actually moving specimens from various locations. And so I'm involved in that right now, which is really interesting. So I'm not sure about sea snakes, but I can look into it for you if you would like. There is the database is called Specify, so it's an electronic database, and then every specimen that's been kind of catalogued in the museum is actually on the database. So I can just inquire, and we can pull out a list and see where we have the specimens, where they're located, that kind of thing. And then, yeah, I mean, the museum, as far as I know, they do sampling trips, but I think it's quite rare just because there's so much work to be done on the collections. And obviously COVID now has been a bit tough for all of us with collections. But they receive a lot of samples. So Dr. Tufik Samai has sent a lot of samples, and people from all over South Africa, actually, because you want to have an institution where your samples are available to everyone in the future. And even if you describe a new species, you actually have to deposit that specimen in a museum. So we have collections from all over South Africa that anyone's welcome to work on. And then with regards to your question about new species and things like that, I mean, I work on sponges with Dr. Tufik Samai, and there's always, you can ask him, there's always work to be done. There's always new species. There's too many. So especially with like your marine benthic invertebrates, no one's really been working on them that much, you know, with regards to taxonomy. So we have groups that just haven't actually been even worked on at all. So there's always scope for work and taxonomy, and then the whole new thing of genetics. So a lot of our older specimens are based just on, you know, your standard morphology. And we need to update everything now with genetics. Yeah.
Sure. So that's I know they mentioned with part of your chat that you do the species pages for the sponges. Is that, you know, are you then rewriting or just adding to those when you do the new genetic testing, and have you had like, so there was a species described, and only now that you guys have actually done the genetics, you find that it's actually falling into a completely different family or genus, and so it's, you know, for all these years, based on the morphological characteristics, we've grouped it here, but in actual fact, now that we've checked, it falls under this family or this group or this order or something.
Yeah, no, so definitely. So the species pages I'm working on, I'm mainly working on sponges, and that's really the problem has been is that you know, we have a lot of species. We have like nearly 300-400 sponges from South Africa. But they're not, that information is not really accessible to any non-specialist. You know what I mean? There's sponge taxonomy can be quite difficult, and it's difficult to identify based just on the external morphology. So you have to look at the internal features, so the spicules and the skeletal structure. So really, we have a huge issue with how to make that information more accessible to like ecologists and even people that are just generally interested, because you need a microscope. So it's really not accessible to everybody. So the species pages, I think, are part of SANBI and the FBRP. So basically all it is doing is looking at the older literature, often which is in French or German, and just putting it into a modern format. So basically I'm just copying out the most relevant information, translating it where I can. Yeah, well, Google Translate, guys, Google Translate where I can. And then but that as well is difficult is that like, you know, with every, I mean, you work on snakes, so you'll know every kind of taxon that you work on has its own terminology. So sponges has a whole thesaurus and all the spicules have different names. So you have to kind of translate that in a way that everyone can kind of understand. And then with genetics, I know Dr. Tufik Samar has been doing a lot of work now on trying to, you know, organize, you can also speak to that, but the genetics of South African sponges. So not much has been done. We're just trying to get into it more now. But yeah, we really need genetics because we have found, so sponges have a very short like larval dispersal in the water column and things like that. And we're increasingly finding, so like on the East Coast now, we're finding species that have previously only been recorded from Madagascar or further in the Indian Ocean. And I think with most benthic invertebrates, we have a huge problem with cosmopolitan species. So you'll have a species that was described in South Africa, but it's also been recorded from the North Atlantic and the Caribbean and, you know, everywhere, just based on the morphology. So now we have to really reinvestigate those and say, you know, are they really genetically the same species? It's unlikely, and we redescribe those. Yeah. So I think Dr. Samar can talk more to that, but yeah, that's what I'd have to say.
Okay. So with those sponges, like some of the species from Madagascar and so on that you find in our waters now, do they hitchhike? So like with cruise ships and other fishing vessels being so active around our coast, and with cruise ships moving, you know, from South Africa and then going up to like Mauritius or to Madagascar, do they hitchhike? So can, in their larval form, just hitchhike on the side of the boat, kind of like a barnacle would, and then end up here and disperse? Do some of them start settling on the boats? Yeah,
so I don't think they really hitchhike in ballast water. So they usually, I think, in the water column for about 2 weeks and they have to settle. So there were some studies, we don't really know much about South African sponge larval ecology, but there's been some studies where they found that we don't really find sponge larvae in the offshore currents and things like that. So previously I was kind of thinking, you know, maybe like other benthic invertebrates, we could have sponge connectivity with like Madagascar because of the eddy, or Mozambique because of the eddies that come down, you know, to find for other invertebrates. We also have the, you know, the strong Agulhas current, which could maybe bringing larvae down. But increasingly they're finding they're not really found in offshore samples. So I think it's more to do, we have a very high level of endemism. The last we checked is about like 50% of South African sponges are actually endemic to South Africa. So I think increasingly we're finding, I mean, there was a new recent paper, I think in 2017, by Tufik, and it described a new Subariti sponge. So it's a huge population off the West Coast. But previously they had thought that it was actually transported, like a Subariti species from the North Atlantic or whatever, that had actually been transported down in ballast water or with the shipping. But then when they did further investigation with genetics and morphology, and they actually found that it isn't, or Tufik found that it's a new species. So we're finding increasingly, you know, the more investigation we do, it's mainly just kind of new species that have been previously. Yeah, and the problem with that as well, we have a lot of our older descriptions are really, really bad. So you'll find someone will describe like a new species in 1931 based off one specimen that they found lying on the beach. So they don't have the color, they don't have you know, it's a tiny fragment, it's desiccated, you know, and then, so now we have to go back and actually re-describe all of those because you can't really compare 100%. And that's where museum collections come in, because now you can go back to the holotype or the, you know, the species that was actually just the specimen that it was described from in the museum, and reinvestigate and kind of redo, you know, or that we're using the modern technology. So you're scanning electron microscopy and our newer microscopes and things like that.
Sure. One last question. Have you, like nowadays, microplastics have become such a big topic of discussion and research. With the sponges, have you ever found accumulation of microplastics in any of the tissues? Or so like when you're looking at the spicules under a microscope, do you ever find microplastics, you know, in amongst that mix nowadays with increases in levels of pollution and so on? Or has it not got into the sponges just yet?
So I'm not sure about microplastics. I haven't seen anything in the samples we've done. But sponges are really good with, they filter feeders. You know, so they're filtering the water constantly. So they can actually build up or accumulate like heavy metals in the water column and things like that. So they can be really actually useful for doing like environmental studies. So around your harbors and things like that, if you can actually look into that and see, you know, okay, we have a buildup of, you know, heavy metals in the water. So they're really good for monitoring in that way. I don't know if Tafika's seen anything with microplastics, but I haven't seen anything personally.
Sure.
No, I haven't seen anything as well. But then, you know, on the other end as well, I think we need to, you know, do some studies on it to see whether, you know, they really accumulate, because there are these filter feeders as well. They've got these nice osteals and oschia. You know, that might just be pumped out of the system again as well. And, you know, some nice experiments will actually be doing with, you know, looking at, you know, just feeding them, you know, microplastics in an aquarium. And then look at, you know, over time period, whether there's anything, you know, on the slides or not as well. The other thing is that to actually catch the water as they pump it out of the oscules, and then just put it through a flow cytometer. And
so what does that do?
So basically the flow cytometer is an instrument that look at microorganisms. So you can actually pick up a shape. It's like a scanner. So you put the water sample through it, and it scans it, and it picks up, for example, small forams, nematodes. I'm just giving you names here. Zooplankton, microzooplankton, and you know, sand particles. And in some of these, they might sit these, you know, microplastics as well. So this is actually one of the projects that has been done currently at our department as well. Actually looking at microplastics in mussels and some other organisms. And then they're actually using the flow cytometer after they've crushed the tissue and all that to check whether there's any microplastics in there as well.
Okay, thank you so much.
Yeah, so we haven't really done it on sponges as yet.
Yeah, I imagine it's an emerging field nowadays. Like I know a few colleagues at UWC that are doing it in seagrass environments and just estuarine environments. Nothing in the intertidal and so on. But I'd always been curious about the type of accumulation you might find, microplastic accumulation among some of the filter feeders in the intertidal. And
I mean, here's an interesting question. What is the greatest contributor of microplastics into the subtidal or a coastal environment?
I would imagine gradual kind of break off and decay of larger plastics that get trapped on the rocky shore or are left on the beach, and then, you know.
Yeah, some of these get into the stormwater drains as well.
Yeah, and then
I'm
walk on the beach. You walk on the beach, you see all those plastic bottles lying in amongst the rocks and so on, and just the gradual pounding from the waves and those sorts of things. They eventually break down, I imagine, and then chip packets and sweet packets and all these things that. But then offshore and in harbors, if you're packaging stuff and you're using those small little plastic pellets and all those things, any pollution.
In actual fact, it's actually car tires.
What?
So basically, when every day people are driving, and the tire gets thinner. Where does all of that go to? Actually, the microplastic. It actually goes into the, it sits there when it rains, stormwater drains, and that is actually one of the biggest contributors to microplastics in the intertidal or marine environment as well. And people don't even think about it, I mean, we've got thousands and thousands of cars going up and down, so yeah.
Sure. If you imagine like that stretch from Muizenberg to Fishhook, all along the coast there.
Oh, we nice to actually look at that as well.
How would you even, if you had to design some sort of sampling protocol, how would you even begin to do something like that along a stretch that is heavily trafficked, like that Muizenberg Fishhook stretch? And like you said, people take it for granted because out of sight, out of mind kind of thing. And how do you even start to sample these tiny particulates that come off of car tires, and the rate at which they come off, and does it vary by brand? Does it there's so many
yeah, exactly.
So many things.
So Fiek, I've got a question from Carl Smith here. He's asking if any national priorities regarding benthic research areas or objectives have been set by either the DFFE or SIRE. I don't know if you know, if you're aware.
Hi Kyle, how are you? Well, basically what we do is that we follow what is coming from the national spatial assessment or national biodiversity assessment, and then we see whether some of those areas in there could be prioritized as well in terms of where we actually do the work. But in terms of prioritizing, we don't have a list. But if we work with other institutions, we find that they've got specific priority areas. For example, Cape Nature will have Table Mountain National Park as a priority. Gokama could be a priority. For some of the offshore stuff that we're doing now as well is to just randomly choose some of the MPAs and go and survey those particular MPAs as well. So every year we have a plan in terms of what we're going to do, either intertidally, subtidally, or offshore. But those priorities can actually change based on the directive that we get from.
Is that Tafiq's connection, or is it mine?
I think it's Tafiq's.
He's gone now.
I was trying to see if any of you were moving.
either provide funds or.
Oh, damn, he's gone.
Well, I think it answered a lot of what I was sort of trying to get to. So I was wondering whether there was, guiding principles, whether it was coming from the NBA 2018 or some other documents. Obviously, as national parks, we would like to try and feed into a national priority rather than on something that would be sort of sidelined. But then the other reason why I was asking is obviously with funding opportunities, whether they link to those objectives, you're probably more likely to get funding if your research objectives and areas are aligned to some of those objectives and to some of those priorities. And then I was just wondering whether there was, because of the new offshore MPAs, the relatively new offshore MPAs, whether there was actually going to be sort of a drive to actually look at those new MPAs as well. Whether there's, not only the offshore ones, but there's also Robberg and.
Yeah. So Carl, so basically, we've started the program in terms of looking at the new MPAs as well as the surrounding EPSs as well. So to date, we've started on the West Coast, and we've did some on the East Coast as well. So on the West Coast, we've surveyed Table Mountain MPA, the extension, Robberg, Charles Bank, Namakwa. We've done Alfred Banks on the South Coast, including the part of the EPSA. We've done Amatoli MPA as well. So basically, we've got another survey in March, and we actually want to go down to Brown Banks and do another MPA as well. In many cases, depending how far these MPAs are from one another as well in the offshore domain, we can either do one or two in terms of the sampling design as well. We did Charles Bank and Namakwa land within 10 days. But the priority for that was just to do video footage. If we extend it, then we can only do one. So we are doing those surveys, and then the Rocky Shore surveys that we've got as well is that we've got monitoring programs within the 4 provinces, and we do that every month. We go to different. We've got 32 sites. And so we try to go to 1 or 2 of them per month, but also collaborate with Cape Nature, Eastern Cape, Isabelo, and the universities as well, to actually allow us to do all of those particular things. One of the areas where we are struggling a bit is the shallow domain in terms of the diving. We've got a number of sites that we're doing, but we're doing jump cameras now, apart from the diving. And then we also have 1 or 2 sites where we have fixed monitoring pins as well, but it's very difficult. So basically, as a department, we choose the areas that we want to do as well as what is priority for us. But like I said, is that if we work with you guys, then, for example, we can prioritize Robberg. We've done Robberg as well. We've done Robberg and Plettenberg Bay area extensively. We had an MSc student on that particular project as well. Okay. So we can actually expand in terms of what needs to be done. And then through the ASEP program, various other projects are run through the ASEP. Gene Harris, them have the Tugela Bank project as well, looking at that particular MPA, especially the paleo reef and or the mesophotic reef structures. And then we have the ASEP looking at the Amatoli as well. So yeah, so in terms of prioritizing, Carl, if you want government to prioritize and work more with you guys, then we can actually try and set that up as well. You know, especially when we're dealing with the intertidal area. Thanks.
Yeah. No, thanks very much for that. I was wondering, can I follow up with one more, Roxanne?
Yeah.
When if you got a new area you're going to, do you guys try and do good bathymetry data, side scan sonar prior to starting with the benthic drop cameras? Or because I'm finding that even in some other MPAs, we've got so little really good benthic data when it comes to your project planning. You actually really are starting in the dark sometimes. And I was just wondering if there was an opportunity to sort of tap into those sort of platforms as well to try and build our bathymetric maps, you know, as a product that we could start to use as a baseline for further research work.
Yeah, that is actually becoming a very important part of the work that we're doing as well. If you look at some of the shallow water stuff for the Amatoli area, we had marine geoscience to map some of the areas as well. For the Table Mountain MPA, we had marine geoscience through Scion when Ali Khots was still here, to map the Table Mountain MPA region as well. I mean, that was probably the best mapping exercise that we had.
Yeah.
And then gene them for the Tugela MPA region as well, is that they just did the mapping now for it, so that we can actually have a better idea in terms of the reef structures or habitats to actually plan our sampling. The problem that we're sitting with, Carl, is that the system that we've got in South Africa only goes down to about 300 meters. So it's a shallow water system that we've got, that marine geoscience has. South Africa has not invested in a deep water system as yet. So therefore it's very difficult to actually do some of the deep water stuff. But what we do as well is that when we actually go out with the ships, we've got these eco sounders, single beam structures, and we make sure that we record from the single beam, and we make sure that we have the ADCP data on there as well. So basically what they can do then is to merge the ADCP data and eco sounder data to actually give you bathymetry maps of an area that we're working at. It's a very, very, very tedious process. There's only one or two people in fisheries that can actually do it. So we've done it with a few of the areas that we've looked at. There's been some talks in government. If there's a new vessel, then one of the features on that new vessel needs to be the deep water system. What we've done, Swen Kerwith, we've got a pole that we can actually link to the Allen Kudzwayo, and we can actually fix it to the Algoa as well, and then get the side scan sonar system attached to it. The other thing that we thinking of doing, Carl, is to actually hire, you know, when we do these deep water sonar work, but then we need to bring out the people from the UK, and they need to have a technician on board as well. So you need to pay the technician, and you need to pay for the system, and you need to pay for the technician on the number of days that he's here, and his flight and his accommodation. So it actually becomes a very expensive.
Very expensive, yeah.
Thing at the end of the day, you know. So, but like you said, you know what, we can't do the work in the dark. It's like going out there and you think there's a reef and it's sand, or you think it's something and it's a ship or whatever, you know. It's and the thing is that we can't spend the time searching for something. We need to go out there and know that it's there and drop the stuff on that reef, because for every minute that we're out there, it costs, or every hour out there, whatever as well, it costs you 250,000 Rand with a ship, you know. So it actually adds up,
yeah,
in terms of the cost allocation as well. Yep.
Right, cool, thanks.
Okay. Anything else?
No, but it's good to see you, Tawfiq, and you, Roxanne.
Yeah, it's been a long time, Kyle.
It's been a very long time.
You're still up in the Gokama area, ne?
Yeah, still based in Sedgefield, yeah.
Still with, but isn't Clovelly's there as well, isn't it?
Clovelly's here, yeah.
Ah, okay. Yeah.
Yeah. Yeah.
So you guys doing a lot of diving as well?
No.
No?
No, not much diving at all because we don't have a supervisor. So, yeah.
Isn't Clovelly a supervisor?
No, you're not.
Not. Okay. Okay. Yeah, that's the other problem as well, eh? I mean, the diving legislation, you know, it's so cumbersome. You know, you need to have at least 4 divers, or you need to have your diving supervisor, you need to have your standby diver and all that as well, you know. And the only thing that you want to go and collect is a sponge, and then you need like 2 people to go with you. Or you can just jump in the water, you know, and just collect the thing. But, I mean, that's, but I presume we gotta follow the legislation as well. So I don't know.
Yeah, unfortunately, yeah, I think it can be a bit restrictive in some aspects. Like we can't even do any estuarine work without having the full team.
Yeah.
But yeah, as you said, we've got to follow it. So. Yeah.
Speaking about the estuarine stuff as well, Carl, is that we've got our director, Gerard Saliers.
Yeah.
He's very much interested in estuarine work as well. So we've got an estuarine program. And a lady by the name of Jabu.
Okay.
She's running part of that particular program as well. She's doing a PhD as well. But it would be nice for you to actually tap in with Gerard and Jabu and see whether we can actually assist with the estuarine work up in the Sedgefield area.
Okay.
And it's always nice to go and find seahorses as well, eh?
Yeah. Not easy to find them.
Yeah, I know, yeah.
Right, cool. Thanks for that.
Take a pleasure, Carl.
There was another question from Matthew just after your and Carl's discussion. I don't know if Matthew, you want to elaborate on your question there.
Yeah, I just heard you mentioned about having some of those technicians on board and how much it costs to have those technicians. Do we have any programs where we train our own, or do we not have the capacity for that yet? Would we have capacity for that in future?
Oh yeah, no, we've got the capacity through marine geoscience. So basically, it's about the collaboration with marine geoscience, where they've got the expertise in terms of, for example, looking at side scan sonar, multi-beam, or whatever as well. It is something that that we can pick up as well. But I mean, they are specifically trained around those particular things as well. So we let them do it. The other thing around equipment, if you look at the ski monkey, Lorraine has been fully trained in terms of the operational aspect of the ski monkey, and also partially fixing it as well. But then we also have other technicians on board that can actually fix the ski monkey if it breaks or whatever. And those are the electronic technicians. And then if you want to go into the domain of an ROV, the funny thing about that is that you need a qualification. You need to be an ROV pilot before you can actually fly an ROV. And that training takes a couple of months as well. And then you can actually go technician 1, 2, 3, supervisor, blah blah blah, whatever as well. So it just depends what you wanna focus on in terms of your expertise. So for example, Lorraine will focus on ski monkey, but you can also focus on, for example, the ROV as well, because it's under systems. So there is capacity and there is training as well, in South Africa. Definitely.
Okay. Thank you. I had one other question, and that was just to do with an increase in citizen science platforms like iNaturalist. Have you guys seen any contributions in terms of just sampling data from those platforms? Anything useful? Or so people.
So you're talking about iNaturalist, ne?
Yeah, and any other citizen science platform.
Yeah, I mean, I'm not a, the thing is that, old school is still with me. I'll leave the TikToker and the Instagram and stuff to Roxy and Robin. But we do post on iNaturalist. Kerry Sink, who's one of the leading marine ecologists, young marine ecologists, deep water scientist, naturalist in South Africa. She does a lot of posting on iNaturalist in terms of the work that they've been doing in the offshore, like posting on cold water corals, or posting on robin sponges, or posting on, these awesome sea fan reefs that you find at a depth of 150 meters or whatever as well. And they make it very interesting, because that is what we need to do, is to actually make our work interesting to, the layperson out there as well. Otherwise, our work don't mean anything. It's blocked into a little science paper. I mean, who reads a science paper? Robin, Roxy, myself, Carl. But you want to reach the millions and millions of other people out there as well. And that is where iNaturalist and the TikToking and the Instagram and not Facebook. Facebook is for old people. And some other stuff, to actually make it out there. And a picture actually tells 1,000,000 words as well. Your people are very interested in these pictures, and you get these awesome pictures that has been taken by these cameras as well. Places that we've never even seen something. I mean, I was amazed that we put down a camera on the west coast of South Africa, and they always spoke about these gobies, goby fish.
The bente gobies.
Yeah, I mean, you get all these papers about the gobies there, the nature paper, but nobody ever saw a goby in the natural environment yet. And here we've got the footage of a goby sitting there, digging in the little hole, and then this little shrimp comes in, whatever as well. And I mean, these are some of the awesome things that you look at, and then these are some of the discoveries as well, and that is why NOAA and all these Smithsonian institutions are so up there, you know, because the first priority is outreach and citizen science to actually play it to the people. Yeah. Cool.
Thank you. Robin, maybe.
Yeah, can I jump in there? Okay, I love our naturalists. So for me, it's a bit difficult because on the one hand, I just love it because I use it to practice my photography. So I mean, I'm not a. I think it's one of those skills, you know, like as scientists, you have to be everything. You know what I mean?
Yeah.
You have to be a good writer. You need to be a good researcher. You need to be able to make images and figures, and I feel like it's just another way of upskilling yourself. So you know, going out, taking photographs. Obviously, it's just fun for me as well. But for sponges, it's a bit difficult. So I have noticed, obviously, it's very difficult to identify some organisms just by images or photos or videos or things like that. So the IDs are not always 100%, but I do think it has value. You know, a lot of scientists, we're very narrow-minded. We think, you know, I think I've heard a lot of people just completely discount our naturalists, and I think, you know, even just the short time that I've been on there, I don't post very often, but there's a lot of interaction, even with the like I say, the community scientists, or, you know, just you'll even get young people who are like, oh, I love this image, or where did you see this fish? And it just creates that interest, you know what I mean? And I've even found like some people, there's possibilities for collaboration on there. And it really is more of a community feeling. So I really like our naturalists. I've also seen, like, you know, it's one of those weird things. Like there was someone, Shame, a young scientist, I think she was working on that invasive green crab. And, you know, it's kind of things like that. So I find if you are starting on work on a certain organism or something like that, and you don't know where to start, especially things like invasive species, you can actually start on a platform like our naturalists. So, for example, I photographed a green crab, I think. So that's an invasive green crab, I think, from Europe, if I'm not mistaken. Or but I found it in Kreeft de Baas, so it's on the west coast of South Africa. And so it's just like that little record might help somebody if you're now doing a research project and you don't even know where to start, what's the range point. So I think taxonomically 100% correct, and I might exclude some of it from, you know, like really detailed place. And for me, I just really started because when I was younger, I mean, I used to spend hours in all the rock pools, and, you know, and then you come back and you can't show anybody what you saw. You say, oh, I saw this amazing wrasse, or I saw this nudibranch, and everyone's like, okay, but you can't show them. And then another thing is having a repository for your imagery, you know, so you can build a life list on there of your species that you've seen, and then as well, it's you know, you have a place. It's you can really only upload if you have you know, your location data, your depth data. So you can go back 10 years from now and I can see a beautiful sponge that I saw. I didn't remember where it was, but now I know it's, you know, in False Bay, around that reef. So if I want to go resample or recollect, I can do that. So I think it definitely has a role to play that a lot of us undermine. And then also, you know what, as well, making science, like Tafik said, making it fun and accessible. So I just found the more that I've been doing, I've been practicing, you know, taking photos of anemones and whatever I find. And then using that in presentations, you know, it makes it so much more understandable for people, instead of, you know, you're trying to go on Google Drive and find a picture of an anemone and then citing people. I just think, it makes you a better scientist, because you're in the field, you know, you want to go out on a Saturday morning and go look in rock pools, or, you know, hike on the mountains. It gets you out there, and it gets you with gets you kind of to that old feeling of science, you know, where you're exploring still. You're actually still exploring. Yeah, that's what I would say.
Thanks so much, Robin. Yeah, I also, for me, the utility was always when I go to a new area. So if it's a beach I've never been to, or a new hiking trail or something, I like to just check iNaturalist so that I get an idea of what species have been spotted there and what to kind of look out for. So you get a nice species list. So yeah, I was just curious because I'm going to get a bit of track here, but with our snake sampling, we found that the number of records and the number of observations has been steadily increasing year by year. So for an organism that was, or organisms that were normally very hard for you to sample and quite cryptic, nowadays we see, you know, you as the researcher might not be interacting with it, but people are interacting with these species every day, basically. And so platforms like iNaturalist, it's just another, like you say, a repository for storing that data. And the quality of the data might not always be so great, but it's still another record of interaction. And as a researcher, it's just another tool to help inform, you know, sampling design or localities and so on. So yeah, I was just interested how you guys would incorporate it into your own sampling.
Yeah, no, so definitely as well. I found, you know, like I say, sponges, it's difficult to obviously identify. So what I've been trying to do now is, if I might take a little sub-sample from like a specimen that I take an image of, and then I can look at the spicules at home and kind of get to a level. But yeah, things like birds, and I'm not sure how you identify snakes really, but I think it might actually be, you know, if you as the expert can, you know, identify to species level, then you know that data is 100% and you can use it in all of your, you know, range extensions or guidebooks or, yeah, and it's building like a community. So it's like, you know, I don't only look at marine stuff. I've been looking at wildflowers in Namakwa, and I go look at fungi in the you know, the magic forest, and it just broadens your skills as a scientist. I think, you know, you start noticing things better. You take, you know, as soon as I go out and I see something new, I'm like, ooh, I gotta take a picture. So my family hates me now. But no, yeah, and again, it's community level. So I mean, when you do that, you feel like part of a thing when you're doing that, you know, our naturalist challenge, and everyone's out there, and also I think it's instigating collaboration. So when you're doing the Our Naturalist challenge and you're organizing a little rock pool tour, you're getting the community actually with the scientists. And I think a big thing as well is the scuba divers. So we have a huge scuba diving community. They're out every weekend, they're taking photographs, but then where's the link? Where's the link between the scuba diver and the researcher? You know, so they in there, they're seeing the new nudibranchs, they're seeing all sorts of ecological interactions, which we can't see because we're not diving as often, because, you know, we're stuck behind a computer, or we're, you know, on a cruise, or, you know, things like that, or stuck in the lab. So I think it really creates those links where you can say, oh, you know what, we get along really well, and you scuba diving every single weekend. Let's try to get you a permit so you can collect these sponges from me when you go to a new dive location. You know, things like that. I think we underestimate the level of collaboration and just instigating curiosity. You know, you have children in the rock pools taking images, and you can talk to them about science. And then again, it's just so much more accessible. You know, when I was younger, I dreamed of having an underwater camera, like absolutely, and they were so like expensive, and you couldn't, you know, it was so unavailable. And these days, you know, with the little, I just use like an Olympus Tough camera, which is also quite expensive now, but I mean, it has a GPS record, a log. You can just literally log your, and I can take little intertidal pictures down to 15 meters, and has a microscope function, so I can take nice pictures of inverts and stuff like that. So I think, I think there's a lot of value in it.
Yeah, I just thought also, like you say, so with us having so many divers and quite an active diving community along our coast, that's just an additional resource to you guys as marine researchers to, you know, kind of piggyback off of their pictures and so on, because planning a diving trip from a research point can be a logistic nightmare sometimes. So to kind of piggyback, like with the snakes, where we piggyback off of, you know, people interacting, the same thing, and you just see an increase in your ability to collect data, whereas you as one person would have collected maybe 2 or 3 samples. Now you've got this extended community doing it for you as well.
Yeah, and I think as well, it's about finding interesting species. So say if, you know, you're working on snakes, maybe you need a genetic sample, but the snake is really elusive. You know, you've done several field trips, which, like you say, it's so costly, getting manpower, getting drivers, getting everyone there. And you can actually go on Our Naturalist, you might find some random person, you know, in the middle of nowhere has seen the snake. You have a starting point. You know, you can go there and do proper surveys. And so I think, it does have value.
Thanks, Robin.
Any other questions from Muhammad? And I see these Qasim Mandova. I hope I said your your name correct.
Actually, I don't have any question, Tawfiq. I just remember we met in Zanzibar a long time ago, doing some stuff with sponges. Actually, I don't have any question.
Okay.
Yeah.
Maybe I must put a face to a name, Muhammad.
Huh?
I say I need to put a face to your name.
My first name is Muhammad and the last name is Muhammad.
Ah, Muhammad.
Yeah.
Are you?
Yes.
Small world, eh?
Yeah.
Any other questions?
Hi guys.
Who's that? Oh, it's Tanya.
Is it just you guys still, or did you have more visitors?
Yeah, we've got enough visitors to keep us chatting along.
Okay, that's nice. We're gonna break shortly for tea, and then we're gonna give the group leaders just time to summarize. So Robin and Tawfiq and Roxy, if anything major came from this room, yeah.
Okay.
Sure.
Must we do that now?
No, yeah, well, now we're going to break for tea, and then we're going to all meet back in the main room at 11. Then each group leader will give a summary, and then Tawfiq, you give a closing. I'm going to send you that little blurb now. It's quite long, but it's fine. I mean, it's been a long week. There's a lot to highlight. And then I've added to your
cool. Okay,
your
fine.
document all the research highlights that everyone has sent me.
Okay, cool.
Yeah. But thanks, it's looking good. But Tawfiq, I will maybe call you during the tea break.
There's a nice. No, it's not a sponge. Something else behind you.
Yeah, I'm like a mermaid today.
Okay, cool.
Cool. Thanks, guys.
Okay.
Cool. Cheers.
What time are we back? After lunch? After tea?
At 11 sharp, I think.
11, cool.
Don't you rush off. I'm going to give you a call quickly. No?
Yeah.
Okay, cool.
Okay.
Thanks, guys.
Okay, thank you guys. I think it's a tea break up until the 11 o'clock, and then we back to the main room. Okay, and thanks for all the interesting questions. Yeah, Roxy, Robin, you want to say anything? No?
Nothing from me, so thanks.
Okay.
No, I just want to say thanks to everyone. It was very interesting conversations.
Okay.
Yeah. Thanks, guys.
Cheers. Bye.
Bye, everyone.